Our company does research on defining the positions of these things called enhancers, which are gene regulatory elements in the non-coding regions of the DNA. So you've got so many genes, but then there's a huge amount of space that's not the coding region, and so there's these regulatory elements. They're called enhancers. But among these enhancers, there's a small subset of these, about 1 to 5%, that are quite a bit larger than the rest. And so if you imagine like a plot of it, it's like an exponential increase for these very large ones, and we call those super enhancers. And so the position of these super enhancers near genes that they're driving tells us what's extremely important for the identity of that particular cell. Now, if that's a cancer cell, that's then flagging some of the key oncogenes, classics like MYC, BCL2, estrogen receptor, HER2, MYB, so kind of the who's list of classic oncogenes. So for my research, we were performing this analysis in a cohort of 66 AML primary patient samples in collaboration with the Magellan lab at Stanford. And so when we performed this profile, we found that a subset of these had a very large enhancer driving RAR alpha. And so that flagged that particular gene as potentially very important for those cells and for this non-APL AML.
Okay. And when it comes to these subtypes, you've got this alpha there. Are there any others similarly associated?
So the RAR, it's called RAR alpha. It's actually a family of proteins. So there's RAR beta and RAR gamma. So RAR beta tends to be silenced or very lowly expressed in cancer. And gamma has some potential skin toxicity. It's been implicated in skin tox, or there's some publications saying that it might help stem cell renewal. So in fact, we're trying not to target those. And so the compound that we're developing, SY1425, is actually a potent and selective RAR alpha agonist as opposed to the other family members.
When it comes to that potency, what kind of sensitivity are you finding?
Retanoic acid receptors naturally bind something called retinoic acid. But our synthetic compound, SY1425, is about 10-fold more potent than that. So you're getting into the sub-nanomolar range. And then really the presence of this super enhancer that I was talking about is predictive for the sensitivity in our preclinical models, which are cell lines or patient-derived xenograft mouse models. And so in the cell line, for instance, where we can get a EC50, which is the 50% concentration of an anti-proliferative effect, we see that about the single-digit nanomolar range for the ones that have the biomarker, but not for the ones that don't.
And could you just talk us through how you are generating the sequencing to identify the markers?
Yeah, sure. So we perform primarily ChIP-seq, which is a chromatin immunoprecipitation with high-throughput sequencing. And typically we look for the histone 3 lysine 27 acetylation mark, which is a mark for these active enhancer areas. And so we do this analysis genome-wide, and we get the positions of all these enhancers across the entire genome. We then perform an analysis to find out which ones are super enhancers, and then we have a bioinformatics team that links those to the target genes that are then implicated as potential targets or the critical regulatory system for that cell. So ChIP-seq. And then we usually do correlated RNA-seq or array-based expression analysis.
And taking a step back, big data as a concept has been incorporated into lots of therapies, and
Sure.
sequencing is playing a part in that. Do you see the two of those coming together in future trials?
In future drugs? Well, yeah. So, I mean, our kind of genomics platform is that we're starting with the primary patient samples because, I think cell lines are good models for testing hypotheses in the lab, but they just aren't quite the same as the primary disease after they've been in a plastic dish for decades, you can imagine. So we try to start with the primary samples. And so the idea is that if we get a collection of those, like the 66 I mentioned in AML, we can do this ChIP-seq genomic mapping across those, pair that with expression, and then get a much better understanding of what the real critical targets are. So, there's thousands and thousands of genes you could go after, but we want to be able to know which ones are critical, and in particular in subsets. And so this serves as both a discovery tool and a potential biomarker for when it's important. kind of contextually.
A personalized therapy from an actual person.
That's right. And so for this SY1425 drug that we're developing, we've actually just gotten approval in the U.S., IND approval to proceed into a phase 2 clinical study. And so we're using a biomarker that was developed to be highly correlated with the super enhancer. So a biomarker that reports on the presence of the super enhancer to select the patients that we believe will may be most likely to benefit from the treatment. So very, very much a genomics coupled with personalized medicine.